Hey guys, in this video, we're going to be talking about enzymes, and we're going to introduce what enzymes are. We'll talk about the active site of an enzyme. Then we'll look at a couple different models for how enzymes function, the lock and key model and the induced fit model. And then we'll talk about the different classes of enzymes. And by the end of the video, you should be able to understand the function and structure of an enzyme, and you should be able to identify the different classes of enzymes. So what is an enzyme? An enzyme is a protein, and it's also a biological catalyst. So a catalyst is something that speeds up a particular chemical reaction. And so enzymes speed up reactions in the body. For every unique chemical reaction your body does, there's an enzyme that catalyzes it. So your body has literally tens of thousands of unique enzymes to catalyze all the different reactions that happen in your body on a daily basis. So there are many, many, many different types of enzymes. And they're very specific, right? One enzyme does one type of reaction. And the names of enzymes typically end in ASE, like peptidase, lipase, hydrolase. And the first part of the name usually tells you the substrate or the molecule that it does the reaction on. And so this protein here in blue would be our enzyme. This little molecule in green and red would be the reactant, or as we call it in enzymes, the substrate. And so the enzyme will bind the substrate, and then it'll convert it into product or products. And the location where the substrate binds is called the active site. Now, the way enzymes speed up a chemical reaction is by lowering the activation energy. So if we look at an energy diagram for a chemical reaction, the products of the reaction are always lower in energy than the reactants, which is why a reaction wants to occur, is because it's a net, lowering of energy level. But in order to get there, you have to overcome some barrier, right? You could think of this like gasoline catching fire. Once it's on fire, it's a spontaneous process, but gasoline doesn't just catch on fire for no reason. You have to give it an initial spark to get it going. Same thing with other chemical reactions. You have to put in a little bit of energy before it's spontaneous. Now, without an enzyme or without a catalyst, there's a large barrier of energy that you have to overcome. With a catalyst, shown here in purple, the energy barrier that you have to get over is much smaller, and so it's easier to make the reaction happen faster. The starting point and the ending point are the same, but you have less energy to put in to make the reaction happen. So it lowers the energy of activation. Now, I mentioned briefly that where the substrate binds in the enzyme is called the active site, because that's where all the activity happens. Let's take a closer look at the active site. The active site is somewhere on the surface of the enzyme, and it's like a little pocket, and your substrate molecule fits in that pocket. But it's not just the fact that it fits the right shape. There are forces going on that hold it into the pocket. And those forces are the intermolecular forces that we learned about chapters ago. You can have hydrogen bonds, you can have dispersion forces, you can have ionic bonds. And these intermolecular forces are occurring between the substrate and the amino acids of the enzyme that point into the active site. So this whole enzyme is made of protein, but right along the active site, there are certain amino acids that are pointed inward. And those amino acids that are pointed inward have intermolecular forces that hold the substrate in there. And so here's just some examples. Your substrate's shown in green. And you could have a hydrogen bond to a serine, or you could have a ionic bond between a lysine and a carboxylic acid, or hydrophobic dispersion forces. So any combination of intermolecular forces can occur and do occur in the active site. Now, here's a actual 3D image of a particular enzyme. You don't need to worry about the specifics, but I liked this image because it's really specific. And so you can see this substrate molecule here in black, and the enzyme is this space filling model that's been sliced away, kind of like a CT scan. And you're getting a glimpse into the active site here. And highlighted in red are the side chains of the amino acids in the enzyme. And those amino acid side chains are interacting with the substrate directly. And it's those intermolecular forces that hold it in there. So it's both shape and the intermolecular forces grabbing onto it. So now that we've talked about binding of a substrate, let's talk about the steps of an enzymatic reaction. So the first step is you got to bind the substrate, right? No reaction is going to happen unless the enzyme and the substrate get together. This binding forms what's called the enzyme substrate complex. So it's the enzyme plus the substrate together as a complex. That's the first step. And then once you've formed the complex, then the enzyme will catalyze the chemical reaction. It will convert the substrate into product and then release it. So binding of the substrate to form the complex. Complex catalyzes the reaction and then releases the products. And once the enzyme has released the products, it's available to go back and grab a new substrate molecule. And so we can think about these enzymatic pathways as cyclical. Enzyme grabs reactant or substrate, turns it into product, releases product, and now it's available to grab more reactant. And it can just keep going and going and going. And so one cycle of grabbing a substrate and turning it into product is called a turnover event. And really efficient enzymes can have turnover rates of like 10 million turnovers per second, which is incredible if you think about it. They're turning over 1 million chemical reactions per second. So incredibly fast. Enzymes are more efficient than any synthetic catalysts that we can make. They're just the best. And so modern biotech actually uses enzymes that we grow in bacteria to do our chemical reactions because they work better than anything we can make on our own. So enzymes are the best catalysts that we know of. Now, let's talk about the specific models that try to describe this enzymatic turnover. There are two models that are used to describe this process. Sorry, let me fix this real quick. That's better. So the first model is called the lock and key model. And so in the lock and key model, the enzyme is sort of like a master lock and the substrate is the key that fits in there and unlocks it. This model was based on the idea that an active site is rigid and non-flexible. And so it only binds substrates that have the right fit, right? So literally like your key fits only your front door lock. So it's a unique shape that fits the corresponding unique shape. Now this describes the specificity of an enzyme. We're like, well, yeah, an enzyme has a unique active site, and so it fits a unique molecule. But that was kind of an oversimplification, and it didn't account for the fact that certain enzymes can actually have multiple different substrates. And so the more modern model is called the induced fit model. And so the induced fit model means that when you bind the substrate, the binding induces a conformational change in the enzyme. It's that the enzyme folds around the substrate more tightly. Kind of like a catcher's mitt when you throw a baseball at it. Like the catcher's mitt is not the exact shape of the baseball, but if it catches a baseball, it folds around it as you're catching it, and it induces a better fit. Enzymes actually work kind of like that. So when the substrate comes in and binds, the enzyme actually will grab on tighter and have a conformational change and induce a fit. So it's the induced fit model. And so this accounts for the fact that proteins, like enzymes, have a little bit of flexibility to them. And it accounts for the fact that you can have substrates with slightly different shapes that both will fit the active site. So enzyme, substrate binds, enzyme changes shape ever so slightly to fit them better, then catalyzes the reaction, goes back to its original shape, and lets go of the products. And we have actually proven that this model is accurate in more recent years. And so here's a real 3D rendering of an enzyme that processes glucose. And so you can see the active site here is kind of the general shape of glucose, maybe not exactly. But once glucose binds, this whole enzyme clamps down really tight on it. And this second image is when glucose is bound. And so these are the actual 3D shapes that we've gotten through X-ray diffraction experimentation. And you can see that the enzyme just completely folds around the glucose really tightly. So it induces a better fit when the substrate binds. So flexible enzyme, substrate comes in, it folds in, grabs it tighter. That's the induced fit model. Now, the last thing we're going to talk about in this video are the different classifications of enzymes. And so there are like, 50,000 different enzymes in your body, and they each have a unique function. So it doesn't really make sense to talk about their names individually because that would be 50,000 different names. So what we do is we group them into categories based on the type of chemical reaction that they do. And so the 6 main classes are as follows. We have oxidoreductases, and they catalyze oxidation reduction reactions. There are transferases, which transfer groups of atoms from one molecule to another. There's hydrolases, which do hydrolysis reactions. Then there are lyases, and lyases either form an alkene or add stuff onto an alkene. Isomerases will take a molecule and rearrange it into a different isomer. Like you can take a cis and turn it into a trans, things like that. And then ligases connect two smaller molecules. So a ligase will take this molecule and covalent bond them to each other. And those are the 6 classes of enzymes. Now, in lab this week, we did an enzymatic reaction using our saliva to hydrolyze starch. And so this was the exact reaction that we were doing in lab. And can you predict the class of enzyme that would catalyze this? And so if we look at it, we know that we are using water to cut or hydrolyze the individual sugars off of the polymer. So it's a hydrolysis reaction. And so any hydrolysis reaction is catalyzed by a hydrolase. So this is a hydrolase enzyme that does it. The specific name of the enzyme, as we talked about in lab, was amylase, because it takes amylose and hydrolysis. hydrolyzes it. Amylase is a type of hydrolase. So just an example for a specific type of enzyme. And that's it for this video. To recap, we looked at the structure of an enzyme, talked about substrate versus product versus active site. We looked at the lowering of the energy of activation. We talked about binding within the active site. We talked about turnovers, the lock and key model, and the induced fit model. That's it for this video. Thanks for watching.